Right, it's a great honor to be welcomed back to this prestigious event, and I'd like to thank the Organizing and Scientific Committee for the hard work that has gone into the meeting and for the kind invitation to talk. I'll be talking about the potential roles of heat shock proteins in the abscopal effect seen with the use of modulated electrohypothermia. I do not have any conflict of interest to disclose. Right. Heat shock proteins, or HSPs, include a large family of molecules, which can be found in almost every living cell. They occur on the cell membrane, intracellularly and extracellularly. Their roles include protection from stresses, regulation of neurodevelopment, and modulating immune functions. As molecular chaperones, they play a role in the protein maturation or degradation, and can either reverse or inhibit the unfolding of cellular proteins in response to stress. They're also involved in the maintenance of steroid receptors and transcription factors. Under certain conditions, HSPs are involved in the protection of multicellular structures and tissues, and may even play a role in systemic processes. Depending on their roles, they may be present in relatively constant amounts or expressed only when needed, for example, in response to stress. Okay, the location of HSPs determines their roles. HSPs found intracellularly act as molecular chaperones and assist in maintaining the balance between the intracellular proteins. They also participate in the regulation of apoptosis and protect the cells from stress. Extracellular HSPs, on the other hand, are important mediators of intercellular signaling. And HSPs found on the cell membrane modulate the membrane characteristics such as fluidity, permeability, and the secretory routes. Okay, the multifunctional behavior of heat shock proteins results in opposing actions. Under healthy conditions, HSPs support the protection of a healthy cell and promote death in dysfunctional cells. And it's these goals that have unfortunately led to the double-edged sword effect in malignancy. HSPs are not able to differentiate between a malignant cell in a system and a healthy cell in a system. They are concerned only with the cell in question. So a malignant cell that is functioning optimally is able to replicate rapidly and cause disease to the system. A functioning malignant cell is protected by the intracellular HSPs. While the intracellular HSPs are protecting the malignant cell, they're enabling the disease and destruction of the healthy host. In this case, a treatment goal would be, for example, to override the protective roles of the HSPs and to promote the cellular destructive roles of HSPs in malignant cells. Most oncological treatment methods cause extra stress, which induces HSPs. Overexpression of specific HSPs provides a selective advantage for malignant cells to inhibit apoptosis and protect themselves from the applied stress. Okay, malignant cells also have much higher levels of intracellular HSPs compared to their healthy counterparts. However, stress also induces translocation of intracellular HSPs to the malignant cell's membranes, where they may alert natural killer cells to the presence of the malignant cell. Once secreted into the extracellular environment, the HSPs may also play a role in tumor immunity. So while HSPs play a role in the protection of malignant cells, they may also be involved in mounting a targeted immune response to the malignant cells. Okay, intracellular HSPs. Apologies for the technical glitch. So the secretion of HSPs on the membrane, or the release of them into the extracellular matrix, appears to be associated with the production of lysosomal endosomes or via the release of exosomes containing HSPs. These HSPs transport intracellular antigenic peptides to antigen-presenting cells, which in turn produce antigen-specific cytotoxic T lymphocytes. This role would be very helpful for the management of malignancies. In this presentation, I will discuss the possibility of using electromagnetic forces to manipulate the location and function of HSPs to our advantage in order to promote the self-destruction pathways instead of the protective pathways. Okay, in order to do this, we need to identify and utilize differences between malignant and healthy cells. One such difference is a cholesterol and sphygmolipid-enriched membrane microdomain, otherwise known as the membrane raft. These are found on the membranes of malignant cells in higher concentrations than on healthy cells. These rafts perform a wide range of roles critical to the cellular function and its interaction with the environment, including triggering intracellular processes. Membrane rafts have a high rate of absorption of radiofrequency current, and in malignant tissue, these rafts may in fact be the highest absorbers of energy. Damage to these rafts places stress on the cell membrane. Chair and colleagues showed that the stress can induce apoptosis. These rafts appear to respond to electromagnetic fields, and these fields may therefore be used to heat up and damage the membrane rafts. Modulated electrohyperthermia appears to induce the upregulation and increased expression of all major HSPs at an mRNA and protein level. Several studies, as you can see here, have in fact shown the effect of MEHT on the upregulation and downregulation of HSP-associated genes, as well as on the development of HSPs themselves. These studies provide insight into the development of the timing of the development of HSPs as well. So here, for example, Schwartz and colleagues showed that the mRNA-associated intracellular HSP peaks at 4 hours post-treatment, and in 4 T1 isograft models. Danix and colleagues subsequently showed that HSP70 folded protein peaks at 8 hours later, which is at around 12 hours post-treatment. This was done in the same isograft model. When we compare Danix and colleagues' results to the results by Andox and colleagues, a similar pattern of elevation of HSP70 is seen in the H29 isografts. Here we see the upregulation of HSP70 in the H29 isograft model treated and peaking at 14 hours post-treatment, as you can see. This is followed by a decline and a return to baseline at 48 hours post-treatment. Andox and colleagues showed that there's a second peak of HSPs that occurs between 72 and 120 hours post-treatment in the H29 models. This is followed by a return to baseline levels after 168 to 216 hours post-treatment. While the first peak is representative of the intracellular HSPs, the second peak is likely due to the release of membrane HSPs into the extracellular environment. This second peak would likely be associated with the pro-apoptotic role of the extracellular HSPs in the immunogenic cell death process. So following that, Bestati and colleagues provided further insight into the localization of HSP70 at 24 hours after treatment with MEHT in the B16F10 melanoma allograft model. Here we see the highest concentration of HSP70 is found intracellularly, with similar levels found on the membrane and extracellularly at 24 hours post-treatment. Yang and colleagues showed that the application of heat stress to HepG2 cell lines results in a massive increase in intracellular HSPs. 3 types of heat stress were applied. They applied heat via water bath, via capacitive heating, and via MEHT. And all 3 samples were heated to 42 degrees Celsius. While all methods resulted in an increase in intracellular HSPs, the increased levels of extracellular HSPs was significantly higher in the MEHT-treated sample, measured at 24 and 48 hours post-treatment, seen represented here with the red line. Andox and colleagues studied the effects of MEHT in H29 models, and in the study, mice were injected in both hind limbs and with the H29 cell line, and only 1 tumor was treated. The treated tumor showed significantly higher levels of HSP90 from between 24 and 216 hours post-treatment. Interestingly, when the control untreated tumor was analyzed, an increase in HSP90 was also seen at around 14 hours post-treatment. So the potential implications of this includes crosstalk between the tumor and immune activation against the tumor. Another phenomenon is seen when we apply the step-up heating. Step-up heating is a gradual heating protocol, and results in the development of more intracellular HSPs in healthy cells than in malignant cells. So a moderate increase in the expression of protective anti-apoptotic intracellular HSP70 was seen in the malignant cells compared to the drastic increase seen in the surrounding healthy cells in this study by Tsang and colleagues. This phenomenon provides an additional opportunity to select malignant cells over healthy cells and renders the malignant cells more vulnerable to stress. The step-up heating also allows for the increased perfusion, resulting in increased drug delivery and increased oxygen perfusion, which in turn increases the cell kill effects of ionizing radiation. So in Tang and colleagues' study, they showed that the rate of apoptosis is also significantly higher during the heating up process than during the period of stable power output. This is suggestive again of the field effect of the treatment on the cells. Okay. So although intracellular HSPs have cytoprotective effects, preclinical investigations have shown an increase in apoptosis in samples treated with MEHT. Cleaved caspase 3 is a marker that indicates apoptosis. It's formed after the activation of caspase 3 during caspase-dependent apoptosis. So caspase-dependent apoptosis is triggered when the intracellular HSPs are unable to restore order following the increased expression in response to stress, and the cell subsequently enters cell cycle arrest. In a study by Danix and colleagues, mice were inoculated with triple-negative breast cancer cell lines. The 4T1 was used, and they were treated with MEHT. The mice were sacrificed at various time points, and tumors were analyzed. HSP expression was transiently elevated with a peak at 12 hours post-treatment and returned to baseline at 24 hours post-treatment. Similarly, the mRNA HSP, which peaked at 4 hours post-treatment, returned to baseline at 24 hours post-treatment. It appears here that the protective ability of HSP70 is exhausted at 24 hours post-treatment and apoptosis begins, because the number of viable cells decreased and tumor destruction ratio therefore increased with a peak at 24 hours post-treatment. At the same time, cleaved caspase-3 expression peaked at 24 hours, suggesting that the decrease in viable cells is associated with an increase in cleaved caspase-3 and therefore an increase in caspase-dependent apoptosis. Andox and colleagues also reported a similar relationship between HSP70 mRNA and HSP70 protein expression after MEHT treatment. Okay, MEHT triggers the upregulation of E2F1 protein in glioma cell lines. This protein is involved in the E2F1 mediated apoptosis. Other facilitators of apoptosis that have been shown to increase following MEHT treatments include PUMA and the P21WAF1. Okay. Other markers of apoptosis, such as caspase 8 and 9, have also been found to be significantly more elevated after treatment with MEHT than after treatment with water or capacitive heating. Okay, the final step in the systemic immune effects of MEHT involve the activation of the immune response to malignant cells. This enables immune recognition and targeting of the immune cells and is the most likely explanation for the systemic effects seen in clinical reports. Vanchik and colleagues showed the infiltration of natural killer cells post-MEHT in a melanoma model. Membrane-bound HSPs are known to facilitate NK activity, and expression of membrane-bound HSPs following MEHT is the likely explanation for the increased infiltration of NK cells. Dendritic cell therapy combined with MEHT also results in an increase in CD8 cytotoxic T cells in squamous cell carcinoma lines. DC therapy combined with MEHT is also associated with an increased concentration of leukocytes, macrophages, and eosinophils in C26 allografts. Okay. The systemic response is reported by the damage-associated molecular patterns. Is supported by the damage-associated molecular patterns, which include the release of extracellular HSPs, the release of calreticulin, high mobility group box1, and adenosine triphosphate. The translocation of calreticulin from the cytoplasm to the cell membrane was observed in the H29 xenografts 4 hours after treatment with MEHT. Vanchik and colleagues measured these markers of damp in C26 allografts. In this graph, we see a comparison between the measurements of calreticulin, HMGB1, apoptosis, DC cell infiltration, and T cell infiltration at 24, 48, and 72 hours post-treatment with MEHT compared to the untreated tumor. It's important to note that the area measuring HMGB1 measured the negative cells, so that is why the HMGB1 appears higher in the untreated tumor. It's actually the negative area that was higher. All right. Immunogenic cell death. This describes the cellular pathway or death pathway triggered by the chronic exposure of damage-associated molecular patterns to the immune system. It's a form of apoptosis that results in the activation of the immune response to dying cells. In oncology, this process appears to promote the immune recognition of malignant cells and anti-tumor immunity and is frequently cited for the major contributing factor to the abscopal effect. Okay, Andox and colleagues analyzed the cytoprotective pathways of HSPs. A gene chip analysis of human lymphoma cell lines showed an activation of the cytoprotective gene network in samples heated with water bath heating at 42 degrees Celsius. The green here represents little to no change, while red represents upregulation. There's a control as well as an MEHT heated sample. Okay, the upregulation of genes such as HSP105, there we go, and HSP90A have been shown to block apoptosis by interfering with caspase activation. In this study, HSP90A was upregulated in the water-heated sample, and the HSPs appeared to play an anti-apoptotic role as they prevented apoptotic death in the cells heated with water bath heating. They did not play the same anti-apoptotic role in the MEHT samples. The ingenuity analysis of the network of HSP-related interactions at a genetic level showed less intensive tumor-protective HSP functions in the MEHT sample. So here we see the BAG3 remains non-regulated in the MEHT sample. As both samples were heated to the same temperature, the difference is likely due to the electric fields. Okay, the abscopal effect is the response of an untreated distant lesion to the local treatment of a primary tumor. Only a handful of radiation-induced abscopal cases are reported each year, and it's generally accepted that the effect is the result of the activation of tumor-specific immune mechanisms. Yoon and colleagues reported on a case of a 72-year-old male with metastatic non-small cell lung cancer. On the pre-treatment PET-CT, multiple regional lymph nodes were visible, as well as a 9.5 centimeter tumor in the right middle lobe of the lung. The patient was treated with 28 fractions of 1.7 grays of radiotherapy and MEHT, as well as granulocyte colony stimulating factor. MEHT was applied 3 times a week after irradiation, and the colony stimulating factor was started in week 3 of treatment. It was administered daily for 10 days. All right, the post-treatment scan, as you can see, showed a good response of the primary tumor and complete metabolic resolution of the regional lymph nodes, which were outside of the radiation field. Okay, here we see the imaging results from a 51-year-old female with breast cancer and symptomatic cardiac metastases. The patient was heavily pre-treated and therefore was treated only with a course of palliative radiotherapy involving 20 grays and 12 fractions. 500 milligrams of capecitabine was administered orally along with 40 milligrams of vinorelbine per week, combined with ipilimumab and nivolumab. The irradiated right chest tumors responded rapidly to the treatment. Interestingly, as you can see here, The non-irradiated metastatic lesions outside the treatment field also demonstrated a sustained response. The patient remained stable for more than 6 months without indications of disease progression. Okay, this next slide shows here Schumacher and colleagues report on the systemic response to localized treatment of a patient with breast cancer and liver metastases. This patient was treated with local MEHT as well as Newcastle disease virus and dendritic cell vaccination. In this case, the patient reportedly survived for 66 months without disease progression and with a long-lasting tumor-reactive T-cell response. This was documented using the enzyme-linked immunospot assay. Okay. Chi and colleagues report on what they believe to be an abscopal response in 3 participants out of a sample of 33 analyzed patients. 3 of the 33 participants were unable to have the prescribed systemic treatment. Case 1 could not have any systemic therapy. Case 2 was only able to receive 2 low-dose cycles of chemotherapy. And the third case was only able to receive immune checkpoint inhibitors. Okay, case 1 was a 42-year-old female with triple-negative invasive ductal cell carcinoma and regional lymph node involvement. She was treated only with radiotherapy to the breast tumor and with MEHT. The bilateral axillary and the left internal mammary metastatic lymph nodes outside the local treatment field showed a sustained partial response. The primary tumor shrank to around 1 centimeter, and the patient was reportedly alive and well 2 years after treatment. Okay, case 2 was a 60-year-old male with a tumor in the renal pelvis. Initially treated with radiotherapy, the patient developed a recurrence in the retroperitoneal region and in the liver. The patient was treated with 2 cycles of carboplatin and gemcitabine, as well as radiotherapy and MEHT to the retroperitoneal region. A follow-up scan showed complete resolution of all lesions, including the non-irradiated liver lesion, and this patient was also reported to be disease-free at 2 years post-treatment. Okay, the third case is a 69-year-old male with cholangiocarcinoma and multiple metastases. He received immunotherapy and palliative radiotherapy to the liver, scapula, spine, and pelvic bone, as well as MEHT. Follow-up imaging showed a partial response of the irradiated and non-irradiated lesions. Okay, and lastly, I'd like to talk about results from our research group. We published reports on a phase 3 randomized controlled trial conducted in Johannesburg, South Africa. The trial investigated the effects of MEHT combined with chemoradiotherapy for the management of locally advanced cervical cancer. HIV positive and negative participants were included. Participants received 50 grays external beam radiation administered in 25 fractions, as well as 3 fractions of high-dose brachytherapy. If the patient's renal and hematological function allowed it, they were also prescribed cisplatin, either 1 or 2 doses during the external beam radiotherapy. Participants were randomized to receive the standard treatment protocols with or without MEHT, and MEHT was administered twice a week immediately before irradiation. Disease progression or disease response was evaluated by comparing pre- and post-treatment PET-CT images, and the post-treatment PET-CT scan was done 6 months after completion of treatment. Okay, we have previously reported on local disease control at 6 months, and we showed a significant improvement in local disease control and disease-free survival at 6 months post-treatment, as well as an improvement in quality of life. What's interesting is that 54 participants in each group had extrapelvic disease on the pre-treatment PET-CT. And at 6 months post-treatment, 24% of the participants in the MEHT group showed a complete metabolic resolution of all disease, pelvic and extrapelvic, despite the MEHT and external beam radiation only having been applied locally to the pelvis, obviously. The frequency of the visualized abscopal effect was not associated with the administration of chemotherapy and was seen equally in both HIV positive and negative participants. So the administration of MEHT also significantly predicted the likelihood of complete metabolic resolution of all pelvic and extrapelvic malignancies or metastases. Okay, we've reported on 2 and 3 year survival, which was almost tripled with the addition of MEHT. And importantly, the participants with the abscopal response seen at 6 months post-treatment remained disease-free at 3 years post-treatment, with the exception of 2 participants whose deaths were not disease-related. Okay. So heat shock proteins have the potential to both promote and suppress the development of malignancies. Being able to stimulate their suppressive roles and inhibit their promotion of malignancy would be a very helpful tool for the treatment of cancer. We propose using the electric field effects of modulated electrohyperthermia to achieve this. We've seen that the non-thermal effects result in damage to the malignant cells, which ultimately leads to the recognition of the malignant cells by the immune system and the development of an immune response to the malignant cells. Applying the electric field appears to excite the receptors on the cell membranes, triggering the release of intracellular HSPs and the expression of HSPs on the cell membrane, as well as into the extracellular matrix. From here, the HSPs are involved in the promotion of caspase-dependent apoptosis and the development of immune recognition. So unfortunately, despite the plethora of literature on heat shock proteins, the precise roles and mechanisms of HSPs in adaptive and innate immunity are still unknown. And there's also still much to learn about these molecules and the effects of MEHT and electric fields on immune modulation and malignant cells. However, we do believe that there is a significant role for MEHT in the systemic treatment of metastatic disease based on these results. So to summarize, modulated electrohyperthermia is a selective heating technique that is enhanced by the electromagnetic fields. It produces a mild heat which is optimal for enzymatic pathways and supports their reactions. The mild heat also supports radiotherapy and chemotherapy. The non-thermal effect results in chemical excitation that promotes the conversion of intracellular heat shock proteins to the membrane-bound heat shock proteins. Additionally, the stress on the cell and the stress on the cell membrane triggers caspase-dependent apoptosis. Apoptosis results in the formation of apoptotic bodies, which act as information signals for the antigen-presenting cells. Once developed, these antigen-presenting cells are involved in the maturation of natural killer cells, and a tumor-specific adaptive immune response is mounted. The membrane-bound HSPs also attract the natural killer cells, which in turn transport the membrane-bound HSPs to the extracellular matrix, where they play a role in the immunogenic cell death pathways. So this immune response promotes the systemic control of malignancy, which is a phenomenon that we have observed clinically as well as preclinically. Okay. Right, I would like to thank everyone who has taken the time to listen to the talk, and I wish you a very successful conference.